Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Okay, so picture this. You get like a little cut, right? And almost immediately your body's like, whoa, we gotta fix this. And these tiny things in your blood called platelets, they rush over. It's like a super fast repair crew.
Yeah, like patching up a pothole before it gets worse.
Exactly. They spread out over the cut and then clumped together to form a clot. Now, for a long time, scientists thought that this spreading out part and the clumping together part were controlled by the same mechanisms inside the platelet. Like it was all just one big process.
Right. It was like, okay, first they spread out, and then as that process continues, they tighten up to form the actual clot. But some new research is suggesting that might not be the whole picture.
That's right. This research from Thomas Jefferson University, led by Dr. Uhasnik, suggests that these two steps, spreading and contracting, are actually controlled by separate pathways within the platelet.
And this is big because it's like a fundamental shift in how we understand platelets and how they work.
And this study was just published in the Journal of Pharmacology and Experimental Therapeutics. So for our deep dive today, we're going to break down this new finding. What did Dr. Naik and his team find, and what could it mean for future medical treatments?
Could this lead to more targeted therapies with fewer side effects? That's what's so exciting.
Exactly. So let's start with the study itself. What were they actually trying to figure out?
Well, their main goal was to investigate whether the signaling molecules that control platelet spreading are the same ones that control clot contraction. Are they both controlled by the same set of instructions, or are there separate sets of instructions for each process?
And how did they go about testing that?
Okay, so they used human platelet-rich plasma, which is basically blood with a high concentration of platelets.
Right, that makes sense. You got to have platelets if you're going to study platelets. So what did they do with this platelet-rich plasma?
Well, they use these things called pharmacological inhibitors. Think of them like tiny wrenches that can shut off specific signaling pathways in the cell. So they would add an inhibitor that blocks one specific molecule and then see what happens. Does it affect spreading? Does it affect contraction? Or both?
Oh, that's clever. So by blocking specific pathways, they could figure out which molecules are responsible for each process.
Exactly. And there's another layer to this. They wanted to focus specifically on what's called outside-in signaling.
Okay, so outside-in signaling, what does that mean?
So platelets have these receptors on their surface, and when certain molecules bind to those receptors, it triggers signals inside the platelet. That's outside-in signaling, signals coming from outside the platelet and going in.
Got it. So the binding of a molecule to a receptor on the outside of the platelet triggers a cascade of signals inside the platelet.
Exactly. So to study outside-in signaling specifically, they had to block what's called inside-out signaling.
Okay, now I'm confused. What's inside-out signaling?
Think of it like this. Before a platelet can even bind to something and start outside-in signaling, it has to be activated first. And that activation process, those are inside-out signals, things that happen inside the platelet that allow it to grab onto a surface and get ready to receive those outside-in signals.
Okay, I think I'm following. So to study outside-in signaling, they had to first make sure that the platelets were already bound and ready to receive those signals. So they blocked the initial activation, the inside-out signaling, using aspirin and apirase.
Precisely. It's like setting the stage, making sure the platelet is already in position, and then seeing what signals control its actions from that point onward.
Okay, so they prepped their platelets, they blocked certain signaling molecules, and then what did they see? What were the main findings?
Well, they found that when they inhibited certain molecules, like phosphonocetide 3 kinase, phospholipase C, protein kinase, C, and focal adhesion kinase, both platelet spreading and clot contraction were reduced. So these molecules seem to be important for both processes, kind of like a general requirement for the platelet to function properly.
Okay, so those are the molecules involved in both spreading and contraction. But what about the molecules that were specific to each process? What did they find there?
Well, that's where it gets really interesting. They found that inhibiting certain other molecules like RAC1 row-associated coil containing protein kinase P38 and MEK has no effect on platelet spreading, but it significantly delayed clot contraction.
Wow, so those molecules are specifically involved in the contraction process, but not the spreading process. That really supports their idea that these are two distinct pathways.
Exactly. And then it gets even more interesting. They found that inhibiting a group of enzymes called CYR-C family kinases actually blocked platelet spreading. But get this, it also accelerated clot retraction.
Whoa, so inhibiting these CYR kinases had opposite effects on the two processes. That's unexpected.
Right. It suggests that SARS-V family kinases are actually involved in keeping the platelets spread out.
Yeah.
And when you inhibit them, it's like you're releasing the brakes and the platelet contracts faster.
That is so cool. It's like these molecules have multiple roles, and depending on the context, they can have completely opposite effects.
Yeah, it's like a complex dance of signals and interactions happening inside the platelet.
Okay, so we've talked about the study itself and the findings. But what does this all mean for you, our listener? Why should you care about these tiny platelets and their signaling pathways?
Well, think about it this way. Knowing that platelet spreading and clot contraction are controlled by separate pathways could lead to the development of more targeted therapies.
Right, because now we can potentially target one process without affecting the other.
Exactly. For example, in some conditions, you might want to prevent clot contraction without affecting platelet spreading. Like if there's a small clot in a blood vessel that needs to dissolve, but you don't want to increase the risk of bleeding. With this new knowledge, we might be able to develop drugs that specifically target the contraction pathway and allow the clot to dissolve safely.
That's incredible. And what about situations where you want to prevent clots from forming in the first place, like around implanted medical devices?
Yeah, that's another great example. Current antiplatelet medications often work by inhibiting both spreading and contraction. But that can increase the risk of bleeding elsewhere in the body.
Right, because you're basically making it harder for the body to form clots anywhere.
Exactly. But if we could develop drugs that specifically target the spreading pathway, we could potentially prevent clots from forming on medical devices without increasing the risk of bleeding elsewhere.
That would be a huge improvement for patients who rely on these devices. It would make them safer and more effective.
It's all about finding the right tool for the job. And now that we know these pathways are separate, we can start developing more precise tools.
Okay, so this is all very promising. But it's important to acknowledge that there are some limitations to this study, right?
Right. This study was performed in vitro, which means it was done in a lab setting using isolated platelets. It doesn't necessarily reflect what happens at a living organism where there's so many other factors at play.
So it's a great starting point, but more research is needed to see how these findings translate to real-world scenarios.
Exactly. And Dr. Naik has said that the next step is to test these pathways in genetically engineered mice. That will give us a better understanding of how these processes work in a more complex biological system.
Makes sense. So we've got the lab results now. We need to see if it holds up in a living creature.
Precisely. And the researchers are very excited about this discovery. Dr. Naik has even called it a holy grail of possibilities for treatments.
He's really optimistic about the potential of this research to lead to new and better therapies.
And in their paper, they specifically highlight the need for safer and more effective antiplatelet drugs. They believe that this new understanding of the distinct signaling pathways could pave the way for the development of such drugs.
So this research is not just adding to our knowledge of platelets, it's also pointing us towards new avenues for treatment.
Exactly. And the researchers also make a really important point about methodology. They say that platelet spreading and clot retraction should no longer be used interchangeably when studying outside-in signaling, because we now know they're distinct processes.
That's a good reminder for other researchers to be mindful of the specific pathways they're targeting when designing experiments.
It's all about precision and understanding the nuances of these complex processes.
Well, I think we've covered a lot of ground today. We've learned about the different signaling pathways involved in platelet spreading and clot contraction, the potential implications for developing new therapies, and the limitations of the study.
We've really gone deep, haven't we?
We have. And now, for you, our listener, a question to ponder. If these two processes that we once thought were one are actually separate, what other biological processes might be more nuanced than we currently realize? Are there other areas where our understanding is incomplete and where a deeper understanding could lead to breakthroughs in treatment?
You know, it really makes you think about the complexity of biology and how much we still have to learn.
Exactly. So keep an eye out for new discoveries in this field and in other areas of cellular biology and pharmacology. You never know what amazing things scientists will uncover next.
And who knows, maybe you'll be the one to make the next big discovery.
That's right. So keep asking questions, stay curious, and keep exploring the fascinating world of science.
Absolutely. There's always more to learn.
And that's a wrap for today's deep dive. Thanks for joining us.
It's been a pleasure.
Until next time, stay curious.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.